The goal of this video is to cover the reactions of the citric acid cycle, with the idea being if we can understand those reactions, we can then be set up to understand how cells use those reactions inside of a metabolic and physiological context. So let's get into this and ask our question, what are the reactions of the citric acid cycle? So recall we're talking about the acetyl-CoA network here. So we've got this molecule acetyl-CoA here in the middle. Acetyl-CoA, we've talked about, can be generated from pyruvate coming over this way, coming out of, say, glycolysis into the center of this acetyl-CoA network. If we zoom in, what you'll notice is that there are actually other pathways that can generate acetyl-CoA, and as well as other pathways that go out of acetyl-CoA. But one of the major places acetyl-CoA can go is into this loop here, which is the citric acid cycle. And what I'd like to do in this video is cover what in the world is happening inside of that citric acid cycle. The basic thing that it's doing is it is fully oxidizing acetyl-CoA into reduced electron carriers. So there are two carbons on an acetyl group. What's going to come out of the citric acid cycle are two carbon dioxides. In the process, we're also going to reduce an NAD plus to an NADH, three of those. We're going to phosphorylate a GDP to form a GTP, and we're going to generate a reduced electron carrier QH2. This is going to happen over a set of 8 reactions, which we're going to go through one at a time. We're going to start up here at 1. We're going to walk through clockwise through each of these reactions to understand what sorts of things are going on. To help guide us as we look through these, I've made what are the sorts of classifications of reactions. We're going to see a condensation. We're going to see 4 oxidations. We're going to see a phosphorylation. We're going to see 2 that are, simple rearrangements. So the overall thing that we're going to be seeing the most of is oxidations, because this is in fact a pathway to oxidize acetyl-CoA into carbon dioxide. So let's just get into these pathways, or these reactions. So the first reaction is a condensation reaction. What we're doing is we're taking the acetate off of acetyl-CoA, and we are gluing it onto a carbon in an existing oxaloacetate molecule. So this is a 4-carbon molecule. We've got 2 carbons coming off of the acetyl-CoA. We're going to glue it together to make a 6-carbon thing. What's happening is at this position here, so if we number the carbons, 1, 2, 3, the 3rd carbon in from oxaloacetate there, we're going to put our acetate at that position. We're going to do so by, this is the methyl here. That is where the reaction is actually going to happen. And we are paying for this, which is actually an unfavorable energetic reaction, by breaking this thioester bond here. So breaking that bond yields minus 31.4 kilojoules per mole, so it's a favorable hydrolysis reaction. Because that hydrolysis reaction is favorable, inside of the active site, we can couple that to, instead of releasing the acetate as a free molecule, we instead glue it onto the oxaloacetate. But that's all driven by this breaking of this thioester bond. Okay, so what have we done? We've gone from a 4-carbon oxaloacetate and a 2-carbon acetyl-CoA to a 6-carbon citrate. What do we do with that citrate? So we take a 6-carbon citrate and we make a 6-carbon isocitrate. Notice we've just changed some of the stereocenters, so it's a pretty minor change. Once we've done that, the enzyme that's involved, or the next enzyme, can recognize the isocitrate and can actually generate alpha-ketoglutarate. So this is a 5-carbon thing. We started with 6 carbons, we end up with 5 carbons. What happened in the middle? Well, we kicked off a carbon dioxide. In fact, this carbon dioxide here, this carboxylate gets kicked off into solvent. It's a decarboxylation reaction. It's very favorable. That favorable decarboxylation reaction is so favorable that it is in fact irreversible. You don't actually see the reverse reaction happening. This reaction is a one-way step inside of metabolism. This is accompanied by the reduction of an NAD plus to an NADH. So we oxidize isocitrate to alpha-ketoglutarate, and in the process kick off a carbon dioxide. Okay, next step. The next one is kind of a doozy. There's a lot of stuff going on, but it uses patterns that we've already seen, so hopefully we can make some sense of it. So in this reaction, what we're going to do is we're going to take a 5-carbon alpha-ketoglutarate, and we're going to make a 4-carbon succinyl-CoA. The first part of this we just saw. We're going to take a carbon dioxide, we're going to kick it off of our alpha-ketoglutarate, so we're doing a decarboxylation reaction, and we're coupling that to the reduction of an NAD plus to NADH. So I'm going to step back slide. That part looks exactly the same, right? We did this isocitrate to alpha-ketoglutarate, reduced an NAD plus to an NADH, kicked off a carbon dioxide. Exact same thing happened here, right? Alpha-ketoglutarate to succinyl-CoA, carbon dioxide got kicked off, NAD plus got reduced to NADH. Cool. But then there's this other wacky thing going on. What's going on with this CoA molecule? Well, it turns out this reaction is so energetically favorable that there's extra energy left over, that instead of just going, say, straight to succinate, there's enough energy to run an unfavorable reaction. It's actually about 36 kilojoules per mole of energy to glue a succinate went the wrong way, a succinate onto a CoA, and yet this reaction is so favorable that it pays for it. So we've basically glued the output of this alpha-ketoglutarate minus its carbon dioxide, we glue it onto the end of a CoA using the energy of this oxidation reaction to pay for it. Okay, who cares? Why are we doing all this complexity? Well, so let's talk about what's happening, and then we'll talk about why we do it. Here it is. I said in words. I should have done it with a picture. That makes much more sense. So here what we've got is our alpha-ketoglutarate and a succinate. It's extraordinarily favorable, minus 67 kilojoules per mole to do this redox reaction. Disappear because we're going to couple this to an unfavorable reaction, and that is the condensation reaction of taking that succinate and gluing it onto our CoA to generate succinyl-CoA. So very favorable, unfavorable. Together, it's still favorable, right? But now we're doing something with that reaction. You might think this is ringing some bells. We actually saw something just like this. This is what pyruvate dehydrogenase does, taking pyruvate and making acetyl-CoA, right? This is the pyruvate here. This is our acetyl-CoA here. It's paying for that by doing a decarboxylation and a reduction of NAD plus to NADH. That's what's happening with pyruvate getting stuck onto acetyl-CoA. Turns out this 8-alpha-ketoglutarate dehydrogenase is evolutionarily related to pyruvate dehydrogenase. So it's doing the same reaction. Going back 2 slides here. This reaction here is just like the reaction of getting pyruvate onto acetyl-CoA, except for it's using an alpha-ketoglutarate rather than a pyruvate as its input. Okay. Why do we do all that? Well, it turns out once you've done that, you can now break that bond to get out some energy and to capture it. So what we're going to do, the next step is we're going to take our succinyl-CoA and we're going to form succinate. That process by itself, just this hydrolysis reaction, is minus 36 kilojoules per mole. We're going to couple that to taking a GDP and making a GTP, so a phosphorylation reaction. That reaction is unfavorable by minus 31 kilojoules per mole. But because we are coupling it to a favorable breaking of a thioester bond, we can pave the cost to make our GTP. So basically, if the previous step was super complicated because we were capturing some of the energy to put it into this thioester bond, now we're getting the payoff from that by breaking that thioester bond to make a GTP. Okay, that was a lot. Let's go into steps that aren't quite so complicated feeling. Oops, wrong direction. Here's a relatively straightforward step. What we're doing is we are in this 6th reaction, we are doing our 3rd oxidation reaction. We are taking a succinate, which has protons implicitly on here, right? And we are oxidizing those 2 carbons. The result is a proton here and a proton here. And we are taking those protons and we are sticking them onto an electron carrier. So we're basically taking electrons and protons and sticking them onto this molecule Q. Now, you might be thinking, I've seen this before, but this was written as FAD and FADH2. And it is true that the actual molecule that is reducing when you go up to succinate is like FAD to FADH2. But it's part of an enzyme complex that immediately turns around and turns this into Q to QH2. And so this is actually only transiently reduced. And so it makes more sense to me that this would be, like talking about an enzyme intermediate as the output. And it's not really the enzyme intermediate as the output, it's what comes out the backside. And it turns out QH2 is this enzyme 60 electrons on that. So in everything that I make, I'm going to try to keep it consistent where Q and QH2 are the outputs of this reaction, not FAD and FADH2. But if you look at a textbook or online, you might see a discrepancy there. Okay. So many reactions in this video, but we are actually getting close. We are on 6 of 8. So in this reaction, we took our succinate, we oxidized it to form our fumarate, and in the process reduced to Q to QH2. Awesome. Step 7 is a very simple sort of rearrangement-y sort of reaction. We're basically sticking a water in and doing a slight rearrangement of the bonds. Simple, not a particularly huge change. That sets up our last oxidation reaction. In this oxidation reaction, we're going to take malate and we are going to oxidize malate. We're going to oxidize malate to form oxaloacetate, and in the process reduce it NAD plus to NADH. This is a fascinating reaction because it's actually energetically unfavorable to do this. It's 30 kilojoules per mole preferring this direction. How in the world does that happen? Well, to do that, we're actually going to do another video. So we're not going to worry about that here. For the moment, we're just saying this is the chemical transformation. We are oxidizing this carbon to form oxaloacetate from malate. Okay. Now, we've seen oxaloacetate before, and what that actually means is we made it all the way from all the way around the cycle, right? So now this oxaloacetate is well positioned to now be have a condensation reaction with an acetyl-CoA, making citrates, and the cycle can then go on and can keep going on. All right, cool. So what we've seen here then is a set of 8 reactions that, if you ask, what is the main stuff that comes out of it, I would say the main stuff that comes out are all these reduced electron carriers. There's a lot of them, right? 3 NADHs and a QH2. You got 2 carbon dioxides that come off. And then this is kind of just a fun one. For free, we get a GTP that sort of gets tacked on. But that's all basically capturing some of the energy that's being generated during all of these oxidation reactions. And then what came in? Well, you had a acetyl-CoA come in and you have a CoA come out. So if you think about this set of 8 sequential reactions, you can actually summarize the whole thing as one giant net reaction that looks like this. You have an acetyl-CoA, 3 NADHs, a Q, a GDP, an inorganic phosphate, and 2 waters that at the end of the day generate a free CoA, 2 carbon dioxides, 3 NADHs, a QH2, a GDP, and a proton. So it looks like a whole bunch of stuff is going on, but most of these things, this one, are going to be immediately consumed by the cell, right? These are something that a cell wants and will use. And so what we're really looking at is I'm going to hide myself, a mega catalyst that takes an acetyl-CoA, generates a CoA, and 2 carbon dioxides. So this whole complicated machine, you could think about it as like a mega enzyme in a sense, right? It speeds up the conversion, the thermodynamically favorable conversion of acetyl-CoA into a CoA plus 2 carbon dioxides. But then along the way, we're going to capture a bunch of the electrons and a bunch of the energy in these intermediates, which are immediately going to be processed in the next steps, which I'll talk about in future videos. All right, so there we have it. That was a tour through the 8 reactions of the citric acid cycle, the set of chemical rearrangements that happens. Big takeaway, it's a bunch of oxidation reactions. In the next video, we're actually going to unpack the energetics of this and then start launching into how it's used. So look forward to that. Thanks for watching.